Hi there, my name is Nicholas Bell, and thanks for stopping by my poster on the role of bioactive compounds in a SIRT-2-in-1 knockdown model of sodium iodate-induced retinal pigment cell damage. I would first like to disclose that I am a PhD student at Lakehead University, and this study has received funding from the Northern Ontario Academic Medical Association. Age-related macular degeneration is a growing concern in the aging population worldwide. It is the leading cause of blindness in people over the age of 50. Two notable studies have investigated the use of vitamin supplementation in order to slow down the progression of dry AMD. These studies had promising results and are available as vitamin supplements in stores today. Our project looks to further investigate the protective mechanism of bioactive compounds with respect to retinal pigment cell damage in a cell culture model. The cell culture line used in our project is the ARP19 human retinal pigment cell line. We use two forms of it, the wild type and the SIRT-2-in-1 knockdown model. We use sodium iodate as a means of simulating oxidative stress and cell damages that would occur in the eyes of a patient with AMD. In order to combat the damaging effects of sodium iodate, pretreatment with bioactive compounds is done before exploring indicators of cell viability and oxidative stress levels within the cells. From the MTS assay, sodium iodate alone induced a decrease in cell viability when pretreated with 15 millimolars or higher concentrations. This can be seen in Figure 2. When pretreated with bioactive compounds of lutein and zeaxanthin in the wild-type cell line, cell viability was increased when compared to sodium iodate alone controls. Beta-carotene pretreatments did not show the same increase in cell viability when compared to the other two compounds. These can be seen in Figure 4. Under the same conditions in the SIRT-2-in-1 knockout model of ARP19 cells, the compounds became slightly more cytoprotective when compared to the control setting cells. When exposed to sodium iodate, the same can be seen for lutein and zeaxanthin with slight variation from the wild-type experiments with beta-carotene. These can be seen in Figure 5. DCFDA assay was used to measure intercellular reactive oxygen species when exposed to sodium iodate. Figure 3 shows fluorescent cell images of increasing concentration of sodium iodate resulting in an increased fluorescence of the probe indicating increases of intracellular oxidative stress within the cells. The intracellular ROS of wild-type and SIRT-2-in-1 knockout cells pretreated with beta-carotene and exposed to sodium iodate was investigated, and we found that the SIRT-2-in-1 knockout cells was seen to have more of a pronounced reduction in intracellular ROS when pretreated with beta-carotene and exposed to sodium iodate when compared to the wild-type cell line. This can be seen in Figure 6. Through these findings, we hope to further investigate the protective mechanisms of these natural compounds on ARP19 cells exposed to sodium iodate and conduct more research to better understand the connection between the SIRT-2-in-1 gene and AMD. Thank you for your time.